I'm Amy Driskell, and I'm on the staff of the Laboratories of Analytical Biology, or L.A.B, here at Natural History. We're on the first floor of the West Wing, and we have labs as well out at MSC in Suitland. So you may not know of us, but I'm going to tell you today about some of the work that I've been doing there for the last 8 years. In 2005, L.A.B was awarded a reasonably sized pot of money to begin a DNA barcoding program. And I arrived in December of 2005 as the project manager for this program. And my job since then has been to establish collaborations, plan, and manage DNA barcoding projects. So what is DNA barcoding and why are people willing to fund it? Well, essentially a DNA barcode is a fingerprint for a species. Basically, it's a sequence that can be taken from anything and be compared to a database and act as an identifier. This is not new science. It's been going on a long time, but barcoding was an international collaboration where we had agreed upon standards and requirements for sequences to be labeled as DNA barcodes. And the idea was we would take the gene, so in animals the gene is the CO1 gene of the mitochondrial genome, and in plants it's a couple of chloroplast genes. We would sequence this gene from as many animals as we could and fill up a database. And these would be high quality sequences. And then these databases, these public databases of barcode sequences could be used for identification of unknown sequences. So in a perfect world, you could have a soup of unknown composition. You could extract its DNA, you really could, and sequence it, and then compare it to the databases of barcode sequences and determine its components. So that's in a perfect world. And of course, the world is imperfect, and this barcode gene that was chosen for animals, the CO1 gene, is not easily sequenced from many large groups of animals, such as nematodes. That's a lot of biodiversity. And it's difficult to sequence out of many different kinds of other organisms, such as animals that eat other animals. So in terms of universality, it's not really that universal. In addition, the databases are sparsely populated in terms of biodiversity. If you have an unknown bird sequence, then you might find a match in the database. But if you have an unknown rotifer sequence, there's no chance. So being part of the museum community, we decided it was our job to fill up these reference databases with as many barcodes for the organisms in the tree of life as possible. So these are the organism specimens that have passed through our hands in the last 8 years. These are just numbers of specimens, not barcodes. You can see we have about 90,000 specimens here. Here are the success rates for sequencing the easily sequenced groups when you have good specimens. It's actually even higher in many of those cases. I can't say that we've barcoded successfully every single specimen on this list because, as I said, many of the groups are difficult to impossible to barcode, although we have tried our best. We haven't given up yet. But in the process of doing this, we have created a very large DNA library of high-quality DNAs that will be permanently stored here and available for future research. So the program has done that. We've had a number of different kinds of DNA barcoding projects at the lab, and I'm going to tell you about two that sort of are different, show you the different ends of the process. The first one has been in collaboration with the Bocas del Toro Research Station of STRI in Panama. Every year we have gone down for their taxonomy training workshops, and as the taxonomic experts collect and name the specimens, we have harvested tissues for DNAs. So in this way, we have had a pretty fruitful collaboration. We've got a pretty good biodiversity sample and lots of good DNAs from freshly killed or still kicking animals. And we hope that this alliance can continue in the future because we don't have any worms, for example, annelids. Another project, wrong way. Another project is sort of tool building, and this has been funded by the FDA. We are developing a method to identify fish fillets with DNA barcodes. And while it's very easy to sequence fish, very easy to barcode fish, what's missing is a complete reference library of fish sequences. So the FDA has funded field trips to the Philippines for the last 4 years. Actually, field trips to the Philippine fish markets, undertaken by Jeff Williams in the Division of Fishes and his collaborators. And in the first 3 trips, they sampled only 1,200 specimens, from which they got more than 780 species. So the fish markets of the Philippines are remarkably diverse. Now, DNA barcoding as part of my program has sort of been declining in years as a major focus, and we have been picking up more and ramping up more to the genomics thing in order to satisfy the needs of many of the curators here at the museum. And you might have heard the word genomics or something else, omics. And exactly what does that mean? And I think it means, in general, we use that word when we are sequencing more of the genome than just one gene, not just the barcode gene, but thousands of genes at one time. And there's a newer sequencing technology which we call next-gen sequencing, next-generation sequencing, which makes it possible to get millions of reads in one run compared to the maximum 96 reads on our more traditional machines. So in last year, Life Technologies gave us a $1.5 million gift, and it included 3 personal genome machines, which are next-gen sequencers. And although it will only sequence your whole personal genome if you're a prokaryote, it still produces 3 to 6 million reads at one time. And this is more than enough data to do mitochondrial sequence entire mitochondrial genomes or chloroplast genomes, which we've done a number of. We have other projects too, including identification tool development and searching for low-cipher phylogenetics. So that's the story of the lab. We're on the first floor, West Wing. We have a lot of equipment. We can do a lot of things, and a lot of people work there. Thanks.
Why are nematodes so difficult?
Because they're the coolest critters. Why are nematodes so difficult to sequence? Well, it's not exactly clear. They're very old. They're very divergent. So it's not clear whether they rearrange their genomes like, say, snails do. But we know that they have a very funny base composition because we've done a couple of mitochondrial genomes. And it's just that the primers that work on other animals for sequencing just don't.